Hello students and welcome to another class with me, and today's topic is immunoprecipitation. Apart from that, we'll also see about co-immunoprecipitation and chromatin immunoprecipitation. Now immunoprecipitation is also there, but we're gonna not discuss it separately because it is already covered in chromatin immunoprecipitation. Yes, ChIP, Co-IP and immunoprecipitation. So yes, please try to understand this concept. And as the name suggests, immuno, that means something from immunology is used here, and that something is we already know. We have already discussed in the previous classes that what from immunology we use the most in ELISA, also we used in Western blot, also we used. That was your antibodies. So yes, antibody will play a role here also. So let's see that what is immunoprecipitation. The first point in our hand is that it uses use of antibodies. That means if let's connect the links. If antibody is used, that means it can bind to some protein, mainly some protein. So yes, if you want to separate, if you want to separate specific protein from a mixture, then you use immunoprecipitation. And what is done is that that mixture contains different types of proteins in it, and when you add antibody, so the antibody will form antigen antibody complex, and the antigen is the choice of protein, and finally it will settle down, and you can just release that specific antigen antibody complex from the whole mixture, and finally you can extract the antigen from there, and then you can study the antigen of your interest. So I can say that it can be used for expression analysis also, like expression of specific protein. Yes, expression of specific protein. Yes, apart from that, as I told you that we're gonna discuss about chromatin immunoprecipitation. So here DNA protein interactions are also seen. DNA, yes. So we are actually writing the applications right now. Yes, we're gonna discuss first the applications, and then we're gonna see the all the techniques so that it will be clear to us. So DNA protein interactions can be seen, and I'll explain you how when we'll discuss about immunoprecipitation. Chromatin immunoprecipitation, and finally protein protein interaction can also be seen. Okay, so all these types of DNA protein interaction, protein interaction, or it may be protein RNA interactions, protein RNA interactions. So for all these, immunoprecipitation is used. Now there are different types of immunoprecipitation. For example, as I have already told you on the first slide, that first is your okay. I'll just discuss the types a little later. First, let's see the principle. So yes, wait a second. Yes, so for principle, as I told you that in the immunoprecipitation slide, I told you that some antibody is used, and it is used to take down a specific protein. So how will we do it? So just imagine that you have a cell lysate, or just let's start with the cell only. So you have a cell, and in the cell we know that many different types of proteins are there at the same time, and I want to isolate this yellow colored protein. Okay, this is my target protein. The first step will be to lyse the cell. Okay, so in a test tube, this is the test tube. Okay, and here is your protein of interest, and with this many different types of protein are there. Now imagine that you have a specific antibody against this protein of interest. So what will happen? This, if I add this antibody inside, so if I add